Okay. Good morning, everyone. Welcome to the CDRI Award Oration. And today, along with the awardees, we have a special guest who is an academic entrepreneur, Professor Vijay Chandru, who is a co-founder and chairman of Strand Life Sciences. To begin the program, I request our directors, Dr. Radha Rangarajan and Professor Vijay Chandru to please come up on the stage. I request the director to present a bouquet of flowers as a token of warm welcome from CDRI family. I would now request you to please address the audience.
Thank you, Neeti. Good morning to all of you. Good morning to our special guests today, the awardees, our chief guest, Professor Vijay Chandru, all of my colleagues, students, members of the CDRI community, family, welcome. Today is a very special day because we are about to recognize scientists who have made outstanding contributions in basic or applied research in the drug discovery and development arena. This is our way of connecting with the larger community of scientists whose interests align with ours. That is CDRI's mandate to develop drugs, diagnostics, and technologies that ultimately contribute to affordable healthcare in India. Excellence can mean many things, but to me, it is epitomized by the ability to do outstanding work again and again. And I'd like to give you an analogy from sports. Many of you heard of Michael Phelps. He's a swimmer, 23 Olympic gold medals. And in the 2008 Beijing Olympics, during his 200-meter swim, his goggles filled up with water. And he basically could not swim with his eyes open. So of the 200 meters, he swam 175 meters with his eyes closed and still won the gold medal. And how did he do that? So in conversations with him, he said that when he trained, he was taught to visualize the swimming pool and count how many strokes it took him to go from one wall to the other, that is to swim the length of the pool. So that day, he swam with his eyes closed, counting the strokes and winning the gold. So that is really the power of being able to do amazing work again and again and again. In Aristotle's words, excellence is not an act, it is a habit. This is what we will hear from, we will hear our awardees talk about their work today. A very brief history of the CDRI Excellence Awards. This was started in 2004, and it is given to scientists or researchers who are engaged in basic and applied research that is noteworthy and has the potential to be developed into product or a technology that can ultimately impact the citizens of this country. Importantly, the work must be carried out in India. We receive nominations from across the country, and the applications are scrutinized by a high-powered committee, which then identifies people who have made outstanding contributions. So this year, we award the Excellence in Drug Research in Chemical Sciences award to Dr. Alakesh Bisai, Professor, Department of Chemistry, ISIR, Kolkata, and Dr. S.S.V. Ramashastri, Associate Professor, ISA Mohali. In the Life Sciences category, the award goes to Dr. Madhika Subba Reddy, Staff Scientist 5, Center for DNA Fingerprinting and Diagnostics, CDFD, Hyderabad, who's still on his way, and Dr. Amit Avasthi, Associate Professor, GHSTI, Faridabad. In addition to our outstanding scientists here, we have a very special chief guest today, Professor Vijay Chandru. First, I want to express my gratitude for Vijay accepting my invitation readily and making this long trip here, despite a very hectic schedule and being deeply involved in the Bangalore Tech Summit last week. Vijay is an icon for a lot of academic entrepreneurs, for entrepreneurs in the life sciences arena. People like me also. He has inspired a generation of entrepreneurs who believed in innovation, who believed in doing this work in India and developing products which were both excellent and outstanding. He co-founded Strand Life Sciences in the year 2000. It was a spin-off from the Indian Institute of Science, Bangalore, where he is a professor of engineering and computer science and continues to be associated. Strand built computational tools to analyze data. And one of their products, Gene Spring, is of course very, very well cited, well used. It was one of the pioneering products to enable data analysis. And then in 2010, it was one of the earliest to build a platform for NGS analytics. And as the company evolved and started to offer clinical diagnostic services, it also became the first NABL and CAP accredited lab in India for genomics and personalized medicine. So Vijay has many firsts to his name. He has been associated with CDRI briefly during 2012, I think during Dr. C.M. Gupta's time. Vijay remembers fondly working with some of our scientists here. I hope we renew our contacts through this visit and welcome him as well as all of our guests who have come here today for this event. Thank you very much.
So it is rightly said that world is small and the world of science is even smaller. A few days ago, I met Professor Vijay Chandru on an online discussion platform wherein he was presenting his views on the upcoming technologies which can be adapted for global healthcare. And I was unaware that in like few days later, I would get a chance to meet him in person, that too in CDRI. Professor Vijay has a vast research experience, as already cited by our director. He has an expertise in computational mathematics. He holds a bachelor degree in electrical engineering from BITS Pilani, and then he did his MS from UCLA, followed by PhD at MIT. He served as a professor at Purdue from 1982 to 1992, and he was working at ISC from 1992 to 2005. His vision to develop technologies for innovation company out of India led to the foundation of Strand's Life Sciences, which is formally called Strand Genomics, and he's co-founder. He served as a co-founder along with Dr. Ramesh Hariharan. His company has captured 30% of global market for analytical software for various omics platform. He has received national and international recognitions. He's recognized as the technology pioneer by World Economic Forum. He's fellow of Indian Academy of Science. And Professor Chandru has served as advisor of Karnataka State Council for Science and Technology, and he is also a member of Atal Innovation Mission at Niti Aayog. He's also participant on WHO's Global Health Foresight exercise on emerging technologies and public health. And his vision is affordable and precision medicine. So medicine for all. With these few words, I would like to invite him to present his lecture.
This is for the pointer. Not required. Thank you. Since I'm wired up, I guess I don't need to actually stand at the podium. So, thank you very much for the warm welcome, both from the director, Dr. Radha Rangarajan, who, by the way, is also considered, an important figure in the whole innovation, I think, ecosystem in India that has been developing. And there's a little bit of an echo. Is that okay? Yeah, sure. You don't hear it. Good. Yeah. I think also the mention by Dr. Niti Kumar of the two of us meeting on the WHO Foresight workshops. I was telling Radha that in some sense we were the two outspoken people on that workshop. I think it's pretty clear that WHO needs more insights from the LMIC perspective and the Indian perspective, and I think we were happy to provide that. As was mentioned, my background is in data science, and while I stepped away from IISc in 2005, I stepped right back in 2016. So I basically had about a 10-year gap when I was running the company, and now I'm associated with both, we've also set up an interesting technology innovation hub at that DST has funded called Art Park at IISc, and I work there as the advisor on healthcare, and basically there we are looking at various ways of looking at data in health and also artificial intelligence applications. We work closely with ICMR on developing gold standard data sets for clinical data analysis. So that's sort of an exciting array of things that one gets to do when you're older and at an emeritus kind of level. So and I'm happy to travel around and meet old friends and visit institutions of great eminence like this one. I'm just going to take about 20-25 minutes to run through some perspectives on both on this interplay between data, precision medicine, but also do a little bit of crystal ball gazing and talk about what the future might look like. And forgive me, I'm not really that involved in drug discovery and development, but so it will tangentially touch on drug discovery and development, but my perspective will be more a kind of life sciences perspective, right? And here is sort of the timeline of interest for life sciences. I think it's probably a prejudiced view, but because to say that everything begins with Mendel is probably not quite right. But But if you start, say, genetics, molecular ways of thinking, that's a pretty good start. And then, of course, you have Thomas Hunt Morgan, who basically told us about chromosomes and the role of chromosomes. And I think, he, of course, came out of Hopkins and did a lot of this work at Bryn Mawr, actually a women's college, sister college to the Ivy League, but in Philadelphia. And then, of course, once he had all these great breakthroughs, Columbia and Caltech and so on grabbed him. But a lot of the early work was done at Bryn Mawr. And then, of course, we go on. I think, I won't try to go through the whole chart, but you can see that there have been very exciting developments, particularly in the last few decades, with the Human Genome Project, and also on the therapeutic side with things like stem cells and cell and gene therapy, and also, of course, immunotherapy as from a oncology perspective. That's very exciting developments. And then, of course, in this millennium, particularly in the last decade or so, this whole development around CRISPR, gene editing, and also, of course, the emergence of these mRNA vaccines during the pandemic, which I think now actually has triggered a certain momentum that we'll talk about a little later. Yeah. And it was mentioned, and my background is in computational mathematics, and in some sense, I'm a data scientist. So I thought I'd also put up a timeline of the data science developments. And by the way, those of us in the business really hate this word artificial intelligence, right? We all want to be intelligent, but why be artificial, right? So we think of these more as intelligent systems and machine intelligence and so on. And actually, artificial intelligence was an artifact that the name was given by McCarthy because he wanted to differentiate from the work that Norbert Wiener was doing. And so I'm basically a descendant of the Wiener school because I basically worked in the labs that evolved from the cybernetics labs at MIT. So, again, very exciting. And what is most interesting is that if you actually put the two together, particularly molecular biology and computing science or information science, it's almost uncanny that these two timelines sort of overlap so much, and there have been so many crossover ideas that this is actually a very nice project in history of science. And I think at some point, I hope somebody, writes probably a Walter Isaacson or somebody like that to sort of pick up this grand theme and make sense of it. But it's pretty uncanny, right? And for me, I feel like I was so fortunate to be born when I was, because in some sense I've watched both these revolutions and in a way participated in both of these. And I think eventually I am a synthesis of both these revolutions, right? And it's been incredible to actually witness this. And so, I think enough history there. But I just want to point out, I skipped a slide. Let me just show that. The other very important thing is that the technology, right? And we often, in science or clinical settings, we don't really also understand, the sort of importance of what is happening in technology, right? And I think it's important to just think that while these might not be thought of as fundamental breakthroughs, the miniaturization, the ability to get cheaper, faster, better technologies, both in information processing, in some sense of electronics, which is governed by what we've been calling Moore's law, right? That's a log linear scale. So actually on the x-axis is time, which is linear, but the y-axis is sort of either you can think of it as cost or processing or compression. There are various ways to interpret it. Cost is probably one good way to just say, what does it cost to build a certain unit of memory or to have a compute of a certain power. And what you see is exponentially it has been reducing. Roughly, Moore's law has been operating at doubling every 18 months. And this has been happening since 1958, when the first semiconductor technologies were created. And so by about 2006 or so, what has happened is that it has entered what we call the second half of the chessboard. So it has actually multiplied 2 to the 32 times, right? When 2 raised to the power 32 is roughly about 4 billion. When a technology gets 4 billion times more effective, you can expect all kinds of magic, right? And of course, we all know, that when I started out as I guess I left my phone somewhere, but when I started out as in computer science with Spilani, we had a computer that, filled maybe half of this room, right? And a mainframe computer. And today, what's on your smartphone is, way more powerful and so on. So I think the impact has been huge. And by about 2006, because we have entered the second half, you almost see singularity, right? You can see it in that graph there. The acceleration is so fast now that everything we thought that would take another 5-10 years is here today, right? So autonomous vehicles, who thought they would be here today, right? And they are, right? And so forth, right? So that's been the power of Moore's law, and we've all experienced it. We've all been touched by it. Now, there's similar law in genomics, and in fact, it has gone faster than Moore's law since 2007, when next-gen sequencing really got industrialized, because that was when the development of the Solexa machine in Cambridge was acquired by Illumina, and then Illumina sort of industrialized it, right? And that, as always in these industry settings, in technology, these things get named after sort of the icons of that industry. So while in electronics we call it Moore's law, who was the chairman of Intel, in genomics we call it Flatley's law because of Jay Flatley, who is now the chairman of Illumina. So skipping forward, I think by about 2010, we are at a situation, and Strand has played some role here. As Radha was pointing out, I think the tools we had built actually helped to annotate the human genome. And by 2010, we were at a point where we had enough understanding of what the genomic code meant and what it would mean in terms of expression of proteins and how those proteins would interact, what would be the pathways, and how that would affect disease, and so on, that we could start making some sense out of just reading the genome, right? And actually have clinical decisions sort of guided by that. And the first breakthrough case was of Nicholas Volker. It was written up as a book called 1 in a Billion, and actually won a Pulitzer Prize. for that story. And I've met, I haven't met Nicholas, but I've met the physician who's there in gear, Howard. He's a geneticist. He was at the University of Wisconsin when this was done. And Howard Jacob. And he, essentially was able to, find a genetic cause for the disorder, and essentially a bone marrow transplant sort of fixed Nicholas, and he's healthy, a college boy, and met his mother about 4 years ago, and she talked about his whole journey and how genomics played this role. So Ramesh and I were at the conference where Howard Jacob presented these results in 2010, and then by then we were already building the analytics, the informatics for supporting next generation sequencing interpretation, but we hadn't actually established our own lab yet. And so this got us excited because, I think we could see that the application was starting to happen. Right? It took us another 2 years to actually find the money and to build our own NGS facility in Bangalore, and then it took another couple of years to get all the regulatory clearances and so on. So, but by 2014, we were in a position to do similar things in India. And Ramesh has written a book called Genomic Quirks, that is published by IISc Press. I would urge people who want to dig in more deeply into all this to read that book. There are 9 case studies of how genomics, the data insights, and then the interpretation and the clinical outcomes. And these are actual real case studies. One is Ramesh himself, who analyzed his color blindness, from genetic causes. So it's really arrived in that sense. And then there are many other examples, as I've put up there, on neuromuscular disorders, and Larry Smarr for what is called participatory medicine or quantified self approaches now, which are becoming more and more part of precision medicine. Just wanted to mention that because of these dropping costs and all of the capabilities, Strand has sort of launched now some tests which we believe can actually help in early detection of monogenic disorders, and the interesting thing is the price point is starting to get accessible in countries like India, right? So this test, I think, is being introduced at about 12,000 rupees, which is still a lot of money, but I think it'll once the volumes build up, these things have a way of dropping. So, but it analyzes 20,000 genes and actually picks up monogenic disorders if the individual is a carrier, but it also is looking at germline related predisposition to cancers, and particularly for women's cancers like hereditary breast and ovarian. This is a way to actually detect this earlier. Now I want to switch from reading the genome to writing the genome, right? I think that it's because I think over the last 10 years, as we were talking about this, CRISPR and gene editing and so on have now brought us into a new era of genetic engineering. And in some sense, this is also a little bit fraught with possibilities of eugenics and so on, and there are ethical considerations, and it certainly requires regulation, and but at the same time very, very powerful. And for people with genetic disorders, particularly the rare genetic disorders, this is really offering a lot of hope, right? And, but it also has huge implications for industrial biotechnology, for agriculture, and for various other aspects. And from a CSIR perspective, I think there are many, many reasons why we should probably also learn how to control this new technology, right? And for those of you who followed the Bangalore Tech Summit, of course, we had Emmanuel Charpentier this year as our plenary speaker, and Jennifer Doudna, of course, is more difficult to get, but her director of the Innovative Genomics Institute, Brad Ringeisen, also spoke at the summit and talked about the applications in agriculture that they're doing with gene editing. So this is a very, again, exciting new technology that we should be tracking. I think finally, I think I just wanted to make this point that I think Dr. Mashelkar always used to say that we need to worship both Goddess Saraswati and Goddess Lakshmi, right? And so we also have to think about what is the impact of all of this on the bioeconomy of the country, right? And if you go back to about 2011 or so, I think 10 or 11, when Obama was the president, yeah, I guess it's 2011, the Battelle Institute had done a study to show that the US had invested about $5.4 billion in genomics, starting with the Human Genome Project and then further on other projects in genomics. And they had calculated that the contribution to the US bioeconomy was about 140 times that, right? So Obama argued with the Congress that, here's a good reason why we have to continue to invest, and then got things like the Precision Medicine Project, which is now called All of Us, and so on, which are been taken forward, right? With genome writing, or with genetic engineering, the impact is likely to be much, much larger, right? And we're, I think Goldman Sachs put out this report where they said in 2018 that the addressable market just in Western markets and just for human health will be something on the order of $4.8 trillion. So these are actually hugely impactful, right? And if we want to go on to be not only hosting G20, but actually, leading G20 in some ways, I think, we have to pay attention to some of these. I want to touch on my connection with CDRI a little bit, and the earlier connection. And here I just want to mention that, we were looking at systems biology in those days, and the problem that we addressed was how to use an approach for combining in silico and in vitro to techniques to actually come up with in vivo predictions of hepatotoxicities. Because this was actually a big bottleneck, of course, in drug discovery and so on. While Strand was able to do the in silico parts on its own, it was not easy. I mean, there were about 10 PhDs, MDs involved, and they worked for about eight years, to actually build the model or the digital twin of the liver. modeling the specific pathways that could capture clinical endpoints with necrosis, steatosis, and cholestasis. So these were the three toxic effects we were trying to model using mathematical systems dynamics models. These were hundreds of differential equations that represented this, and you could run simulations. However, to understand how the drug first metabolizes, we realized we couldn't do that in silico. And so we actually worked with CDRI in conceiving how we might actually build assays to capture how those metabolic rates and so on would be set up, and then that would be plugged into the model and the simulations run. This actually led to a platform which was quite good, and pharma companies were paying us about $10,000 for testing a molecule, right, through this whole platform. And once we established that there was a market, the Biocon group said that they would be interested in acquiring it, and so we passed it on to Syngene because this was not a kind of CRO business that Strand was adept at building, so now it is part of the Syngene offerings, right? So the team and the lab and the technology, the IP, everything was handed over to Syngene. And so I think it's also an interesting story to tell that how CDRI Strand first developed this R&D, translated it, and then it's finally gone into full commercial sort of operation. So I think it's hopefully it'll get written somewhere in the annals. Then let me also just, of course, I guess this is very much in all our minds that something extraordinary happened during the pandemic. For the first time, vaccine was developed from knowing the pathogen to actually having the vaccine in less than a year, and that will be considered one of the significant developments. And as we improve our understanding of immunology, I think there's going to be probably a lot of vaccine development work that can go forward. And this is where I feel that there's an emerging, again, a blending of mathematics and biology and and systems modeling, which I've called systems immunology. I think we'll probably see some very exciting developments in this. I think this is the crystal ball gazing. I think what can we expect in 2047? And I guess we are in our 75th year of independence. It's natural to think about what happens on the 100th year. And I think, of course, the Prime Minister has been setting out all kinds of visions on the economy and so forth. But I think from our own little perspective on life sciences and its impact, and I call it, we are going from digital technologies to living machines, right? And I think we are sort of seeing a huge impact of that. Just put down some thoughts there, but I think we will see how much of this pans out, I guess. But it's certainly good to have these stretch targets in mind, and I won't try to go through all of that, but I just wanted to point out that the impact goes beyond vaccines, diagnostics, and treatments. I think we are also, I think food security and environmental security and various other, and there's also a reverse impact that computing will actually benefit from life sciences, and we already know that DNA storage, data storage, and DNA computing and so on are possibilities, and these will be developed further. Particularly storage, I think, is going to be definitely an impact. And let me just end by saying that, you know, I have to be very grateful to the institution, the Indian Institute of Science, which allowed, I think, this experiment. We were the first academics, I think, with the sanction of the university, to actually set up companies. We actually had two companies. One was the Simputer, the handheld computer, and the other was Strand. And I think, and you know, the fact that there were four of us together actually allowed us to be a lot more courageous because I think, you know, entrepreneurship can be a lonely journey, and I think having a gang of four really helped a lot. And that's Ramesh and me on the right, and yeah, Vinay and Manohar on the left. So all four of us were involved in both companies, but Vinay and Manohar really led the Simputer, and Ramesh and I worked on Strand, and then we sort of helped each other in many ways. I think after that experience with Strand, I'm now involved with a whole bunch of companies. So I've sort of created my own little deep tech ecosystem, as I call it. And some of these are exciting. I think I just wanted to mention a Lantern, which is doing repositioning of cancer drugs. Hanujan Therapeutics, which is developing for Duchenne muscular dystrophy, you know, antisense oligotherapies. And CRISPR Bits is our new group that is looking at various uses of CRISPR technology. Right, I'm done. Thank you.
So we have time for two quick questions. Anybody from the audience? Anil.
Sir, in this whole journey of your academician turning to the entrepreneur, what are all the problems, real problems, which you faced from the both sides, from getting this transition done?
Right, yeah. So, you know, I think this entrepreneurial journey, by and large, is not for the faint-hearted, and it's, and particularly in life sciences or health sciences, I would say, you know, you really need to be brave. I think emotional IQ is what is most important, right? And no, there were challenges, huge challenges. I think with obviously there were many academics at IIC who just felt that this was a waste of time, and you know, and also probably that I was misleading Ramesh Hariharan because he was a really bright young academic star, and you know, all kinds of issues. But let's leave that aside. I think the leadership was very supportive. The council of the institute was very supportive. Professor Raja Ramanna said, you know, this should happen. Professor C.N.R. Rao was very supportive. Govardhan Mehta, you know, at the top, everybody was supportive. So it happened, right? And I think, you know, from the industry side, you know, it's, you know, those of us who have done this, you know this, there are days when you think you won't have an enterprise the next day, right? I mean, I think there were at least 3 occasions when we felt we were collapsing, or, you know, it would not survive. And as I said, I think it really helped to have partners because, you know, we could sort of somehow get over the tension, right? And so, you know, you'd have to put on a brave face because you didn't want, you know, the rest of the company to know, you know, the challenges in some sense. You'd have to put on a brave face with the family at home. But, you know, I think it, so living with that little bit of tension is certainly the challenge. But I have to say that, you know, my academic training was actually quite good for becoming an entrepreneur, right? If you think of yourself as an active academic researcher, you raise money, you market your work, right? You lead teams, you manage people. Right? What else do you need for running a company? You just need to do it faster. You have to do it in a much more focused way. You have to answer the phone when it rings, right? Stuff like that, right? But otherwise, your training as an academic is actually really quite good to becoming an entrepreneur. So let me leave it at that. Right, thanks. Right.
So one last question, Monica.
Just a quick query, sir. Wonderful talk. And my query was around the genomic diagnostics you were talking about, particularly in the cancerous cases. What I believe most of the time, these diagnoses are based on the Western population. So
Right.
when you are doing it, how you are incorporating the new ones, it's coming in the Indian population. And I believe it should bring the cost down. So what do you think about it?
No, absolutely. And I think the ethnicity factors, and we were talking about it just this morning, I think are huge. I think, for example, Strand has now done maybe about over 20,000 cases of hereditary breast and ovarian cancer diagnosis using NGS. So we actually have an extraordinary database, in fact, and an understanding. And because we built the entire informatics infrastructure that is used for analyzing the next case that comes along, we actually feed this data back. And we actually keep learning from the markers that we are seeing in the Indian population. So what we are building is actually sort of focused on South Asian predispositions and so forth. And by the way, I only talked about germline. On the somatic side as well, I think, again, ethnicity is important. And again, I think we are building a large repository. And these are why, the India Genome Project and the CSIR, TCGA, and so on, have been proposed as projects and are being executed because, we need to build this understanding better. And I think the cost coming down is it's a combination of things. I think the technology itself is getting cheaper faster. I think, I think what Strand has done is we have taken out the cost of the informatics in the sense that we have built in so much automation, right, that essentially the expert human involvement is only at the finishing steps of checking the reports, right? A lot of it is generated. I again dislike what IBM tried to do with Watson and to claim that everything is going to be done by Watson. But Strand actually has something like a Watson. We call it Ramanujan. It's actually internally used for doing that level of automation in analysis. So essentially, as the data comes out, it's a single click button that will generate the variant call files. The variant call files go into the cloud, and then there's a whole bunch of sieving techniques, and you narrow it down to the markers that probably are important. All of that, a lot of that is done automatically, right? And then in the last bit, there would be an expert to look at the data. So that brings the cost down.
So thank you again for sharing this wonderful and inspiring journey. I request our director to please come up on the stage and present the token of appreciation from CDRI family. So can you please stay here as well? So a few of our awardees would be leaving early, so we want a group picture of all the awardees with Professor Chandru and Dr. Radha Randarajan. So I request Alakesh, Ramasastri, MS Reddy and Amit to please come up on the stage. And I, again, a warm welcome to all the awardees. Congratulations. And I request the director to please present a token of warm welcome to all of them. Yeah, all our awardees, yeah. So, Alakesh. Thank you all. So now we will begin the CDRI Oration Awards. Thank you. So in the interest of time, housekeeping rules, the buzzer will ring at 20 minutes. So, so the first speaker is Professor Alakesh Besai, and he did his PhD from IIT Kanpur, and after that he did his postdoctoral research at UC Berkeley. He relocated back to ISER Bhopal, and currently he's working at ISER Kolkata. Professor Alakesh's research contributions have been recognized by national and international agencies. He's a Young Scientist Award. He's received Young Scientist Award from CSIR and DAE. And his group has been involved in development of technologies and strategies for target-oriented total synthesis for complex alkaloids of biological relevance. So we'll look forward to your talk.
Hello, is it on? Good morning, everyone. Thank you, madam, for the kind invitation. And sir, it was a great lecture. And thanks CDRI for the award. And so I'm going to talk about our research, which is actually based on the chemistry, and you will look at many structure in it. Okay. So if you see, my title of my talk is the synthesis of biologically active natural products and valuable drugs for human well-being. So I will particularly talk about the natural product and its connections with the drug discovery program. So the motivation of our group is definitely CDRI Lucknow, where Central Drug Research Institute play key role in the development of basic science into the drug discovery. But what our group thinks about this natural product and drug discovery in the very simple way, in chemistry way. So what we thought that nature is nothing but a big laboratory where nature is making many molecule, breaking molecule, and based on the need. And the chemist would like to sort of adopt some of the strategies made by nature and then do the testing in the laboratory. Okay? And if you look at the ADME principle, where everywhere you will see the chemistry play bigger role, in nature, in biology as well as in chemistry. It actually tells us why heteroatom in drug molecule is very, very important. And if you look at the distribution again, the simple principle, chemical principle, homo-lumo interaction, that is very, very important. And this is what the most important part, metabolism, and that is where actually the drug discovery sometimes fails. Okay? We'll come to the point, because why nature is so smart? Because look at how nature makes molecule and break molecules. If you look at the synthesis of glucose, like through fructose, you can see the nature can make it and break it, based on the principle which is known as principle of reversible, microscopic reversibility. Similarly, you can think that fatty acid synthesis and degradation, it is one way. It is actually synthesis and degradation both has to operate. Okay, and that is how the basic idea of principle of microscopic reversibility came into the picture. And then of course excretion. So basically, if you look at all drug molecules, they should be chiral. So therefore, the stereochemistry is very, very important consideration. And in fact, I will show you some of the natural products which are actually the drug molecule which are available. If you look at the anti-cancer drug, doxorubicin or danorubicin, they are nothing but natural product actually. Nature makes them through polyketide biosynthesis. Okay. So now the natural product also becoming the inspiration for the discovery of drug molecule. If you look at folic acid, okay, which is a nice drug molecule. Now, if you look at the anti-cancer drug methotrexate, it is nothing but a folic acid inspired molecule where N-methylation and followed by amination over there leading to anti-cancer drug. So here is another molecule where it shows that halaven, very, very active, which is again inspired from natural product like helicondrin B. And in fact, the truncated one, if you look at the right hand side, this part is only responsible for to act as anti-cancer agent, and look at the dosage only 1 milligram. So in this connection, I was looking at the contribution from the CDRI, and if you look at that art ether, it is nothing but a natural product derived drug. Okay, it is, you all know, 2015 Nobel Prize. So Professor Yu got the 2015 Nobel Prize, and for her work on artemisinin. But if you look at this RT ether, is nothing but it's a reduction of lactone followed by addition on ethanol. Okay, that leads to alpha and beta. This shows actually beta RT ether. Again, if you look at the quinine, that is another drug, which is commercialized. And if you look at the chemical component for this drug is nothing but bacoside. Okay. Now bacoside, if you look at this, is nothing but it's a sugar unit. It's connecting with this epimeric position. Now if you look at this, these are nothing but terpenes. It's a functionalized terpenes. Okay. In fact, this is what is the responsible, for this acts as Alzheimer's drug. There is another one, gugulipid. So if you see the gugulipids, it has again a lot of mixture of compound organic molecule, and one of them is like this. Now if you look at this molecule, this is nothing but this is a tetracyclic terpene molecule with lot of functional group like secondary alcohol, tertiary alcohol, and acetate. Okay, so this is another drug actually commercialized by CDRI. So the point I am trying to make here is that if we follow nature, nature can act as the best guide for the drug discovery. Okay, because all these molecules are having three-dimensional architecture. Now, our group have been developing various strategies for carbon-carbon bond-forming reaction as well as carbon hetero bond-forming reaction for the synthesis of many biologically active molecules. Like if you look at this physostigmine, this is used as Alzheimer's drug, which is actually commercialized, and we have shown the strategy for the synthesis of formation of this benzylic all carbon quaternary stereogenic center. And if you look at the other natural product, which is SN methyl, and if you just do demethylation followed by reaction with this isocyanate, that can give rise to physostigmine. Okay. Along the similar line, we have also developed strategies for the synthesis of lysergic acid derived natural product. Okay. These are alkaloids, cycloclavin as well as clavin alkaloids as well as lysergic acid. Now, you all know about LSD, that is actually banned because of 1960s counterculture in western countries. What is LSD? If you see this portion, if you have NN diethyl carboxamide, that is nothing but LSD. So, what is that? That is nothing but a synthetic drug molecule from the natural product. Okay. So, what we could do? We could actually synthesize this lysergic acid and we can actually couple with this amino alcohol. So, very recently we have shown that galanthamine, which is nothing but amide delisi alkaloid, and we have shown the synthesis of this molecule in a stereoselective manner, because there are a lot of research based on galanthamine and other molecule, because this is also another Alzheimer's drug which is commercially available, and people are also doing a lot of studies with the derivative of galanthamine. We have seen in the literature that there are other alkaloid which are actually having atrobisomer that is leading to this connection of this kind of biaryl, and we have made the strategies for such kind of molecule, and this is going to be published, but these are the molecule we have already published in the literature. Can you please give me the water bottle? I have one water bottle there. So, this is a slide where actually I have summarized the kind of natural product we have been working on. Thank you. So, if you look at all these are nothing but having lot of heteroatom anchored molecule like nitrogen and oxygen, and these are actually isolated from nature from various species. So, these are the structural scaffold actually we have been working, and then we have developed their strategies for their synthesis. I am not going to discuss about all this. I will only take 1-2 example towards the end. These are other type of alkaloids, those are actually isolated from nature, particularly lysergic acid and their derivative, as well as complex alkaloid like chimonanthine and calicanthine. Okay. So, what we do is that we look at the biosynthesis and then the way nature make them. So, we try to follow the biomimetic synthesis. So, what we do? We try to make them fast and then do some sort of acid catalyzed processes to make this kind of complex structure. So, of course, there are other complex molecule also isolated from nature. Those are having diketopiperazine based pyrrolindolene alkaloid. So, here is what the proposed biosynthesis of calicanthine from chimonanthine. So, in the presence of acid, this ring can actually open it up, therefore, it can help in rearranging this structure leading to the complex isomer of this molecule. So, what we thought? We thought of having a unified strategy for many natural product actually shown like this. So, if you look at these are all isolated from nature. So, what we thought? A common strategy for this kind of molecule will actually give us the opportunity to access them in significant quantity for biological evaluation. So, therefore, we have devised strategies where we can get this kind of diol tetraamine dialdehyde, and which can undergo actually cyclization leading to a number of natural products at one go. So nature makes actually them from the tryptophan oxidation of this tryptophan leading to this kind of dimeric structure. And when the dimer, since there are 6 stereogenic centers, you can see that a number of diastereomeric possibilities would be there. However, at the end, a PLP mediated decarboxylation, if you decarboxylate them, that would lead to the this kind of simple structure from this kind of dicarboxylic acid. And so based on this hypothesis, what we are doing is that we are taking L-tryptophan and cyclizing it, oxidizing it through electrochemical oxidation pathway, and then we are using this decarboxylase enzyme. So basically we are using a PLP derivative. Therefore, we can do this decarboxylation leading to this kind of alkaloid synthesis. So basically what we do, we try to first look at the literature and see their biosynthesis pathway, and from there we try to develop strategies so that we can actually get a unified strategy to access a number of alkaloids. So these are some other complex alkaloid which we have synthesized. It is yet to be published, but if you look at the biological activities, these are having a lot of interesting bioprofile, and even by just changing this R group, we can actually able to develop a lot of new molecule which are actually which could be taken up for biological evaluation. So this is another one isolated very recently. So urocristatin, if you look at urocristatin plus and minus, again they are having a dense array of stereogenic center. What we try to do is that we try to make the diketopiperazine arising for L-tryptophan as well as valine, and then we do at one pot, we try to do single electron transfer mechanism to do this carbon-carbon bond formation followed by cyclization. So at one step, we can actually get a diketopiperazine getting dimerized leading to this kind of structure. So here are some of the other alkaloid like indolosesquiterpene alkaloids. So if you look at these structures having again 4 contiguous stereogenic center, in addition to these, there are like carbazole motif. Okay, and carbazoles, since this kind of nitrogen actually can stabilize a radical because of the because as there are 2 aromatic ring flanked between this, so basically it can stabilize this radical. So therefore they can give rise to a lot of different type of dimerized product like digiamycin C. If you look at this, is nothing but dimer of this. Okay, not only that, there are other natural product like diterpenoids. They are also isolated. So what we see that a unified approach can be done for both these type of natural product following a common strategy. So you can see there could be NN bond formation leading to atropoisomer. Like if you look at binol, R binol and S binol, there are, you can see that a carbon-carbon bond is leading to the atropodiastereomer. However, this is the first example in the literature showing that a nitrogen bond formation also could lead to the atropodiastereomer. Okay, and so how nature is making it? So basically it is a N-center radical which can get stabilized at various position of this aromatic ring, and therefore they can do combination. So if you have a monomer, they can be actually oxidized to get NN bond, or it can, the same precursor can also give rise to other natural product like digiamycin C. So to make this dimer, we had to synthesize this monomer, and these are the some of the strategies reported in the literature, including there is another strategy from my postdoctoral advisor, Professor Richmond Sarpong from University of California, Berkeley. Also from our country, my friend Dethe from IIT Kanpur also shown a very nice strategy for this kind of molecule, because these molecules are considered to be a nice drug even for the treatment of COVID. So this is where the strategies are reported. I mean, these are the summary of the 4 strategies. Those are reported. Also, in addition to that, Baron et al. Showed the first synthesis of digiamycin B And interestingly, what they tried to do, when they tried to do dimerization of this geomycin, they could find that only digeomycin B can be obtained, and they did not obtain even any trace of digeomycin A. So this is where actually we ask a question that if these are atropodiastereomer, why not the other isomer? What happened to this other diastereomer? And that is where our interest came in this area, and we tried to explore. So if you look at the other congeners of these alkaloids, these are very recently they have been found, as a potential drug for the treatment of COVID. Therefore, we thought of exploring our chemistry to access all these natural products at one go. So what we thought? We thought that all these natural products can be actually obtained from a common precursor like this kind of diketone coming from here. So I am not going to discuss this chemistry in details, but what we have done is that out of all these 4 positions, we tried to very selectively do CH activation of aliphatic system. Therefore, we can functionalize this ring. So if we can functionalize, that can be utilized. So what we did is that we tried to oxidize this molecule to get iodo or bromo over there, and then initial attempts were rather very frustrating. However, upon prolonged this optimization, what we could see that even PIDA iodine did not work, which has been used by Phil Barron for his synthesis. But what we could see that just using NBS and catalytic amount of acid and in acetic anhydride, we could actually follow this step to get functionalized skeleton. And this is actually going through, we believe that it is a stepwise mechanism where actually we generate a carbocation, and that is doing 1,2 migration. Again, it is going back 1,2 migration, leading to a stereoselective synthesis of this bromo compound, and which was in fact, this compound, isolation of this compound actually showed that this is what is the pathway. Then what we did, we just took this molecule, and as per the biosynthesis, we have just oxidized it to get NN bond. And so here we have shown that both atropodiastereomer can be synthesized in the same pot. So I am not going to discuss about this molecule. It's a very complex one. It's not yet reported at all, not yet reported in the literature. What we have shown that a pericyclic reaction can be utilized for making this kind of molecule. What we did, a 4+2 cycloaddition followed by a 2+2 cycloaddition we have utilized, and what we have shown that both partner can be synthesized from again a common natural product. So basically a common natural product has been explored to get both these partner, and which under Diels-Holder we got a pair of regioisomer which was oxidized, and then chromatography separation gave us this molecule, and this upon treatment with light it gives thioiodide J, and which was confirmed from the crystal structure. This is a very recent result, and we are very excited for this one to be published. So in conclusion, what I try to say is that natural product and drug discovery has very nice connection, and in fact in the future we believe that there will be a lot of synthetic drug will be replaced by natural product, because of what nature is following that principle of microscopic reversibility, because making and breaking should happen, because everything happening is nothing but single electron catalysis. You are listening to me because of single electron catalysis, or I am looking at to you, everything is nothing but electron flow. So therefore, natural product in drug discovery will have very good success, and then we also try to see that natural product can be synthesized following a bio-inspired pathway, and of course asymmetric catalysis also is very important. With that, I would like to again thank CDRI for this award and for this encouragement, and I am thankful to my institute, CSIR Bhopal, where actually I started my career, and I was there for 10 years, then recently moved to CSIR Kolkata. I am very happy to all these funding agencies, and happy to CSIR for their generous support, and also happy to my collaborator and student for their all this good work. Thank you very much for your kind patience.
Thank you, Alikesh. It was really nice piece of work, and you showed how by making these adopting different methodologies, we can have complex molecules and nature-inspired molecules.
Yeah.
So we have question for one quick question from the audience. Yeah, Kishore.
Alikesh,
You're audible.
okay.
Yeah.
So how efficient and selective is that NN radical coupling reaction? Oh.
Yeah, so basically, if you look at, there are only, the yield was 39% and 23%. So if you look at it, is approximately 1.3 to 1 DR, right? So the selectivity is not that great, and selectivity is coming from because of this three-dimensional structure in that cyclohexane decalin system, okay? So frankly speaking, there is no selectivity. However, this process showed what nature makes these two natural products. Actually, it is a NN coupling.
It's a nice reaction. Yeah.
Yeah.
Thank
Okay,
you.
so if you have further questions, you can have discussion with him during the tea break. We'll have quick 10 minutes tea break. Because of the renovation of the dome, tea arrangement is done in front of the KRC. Yeah, and we come back in 10 minutes, and then we'll have Amit talking about his work. Okay, great.
you.
Okay, welcome back. We'll quickly start the session. Now I would call upon Dr. Amit Avasti from THSTI. Dr. Avasti did his PhD from NCCS Pune, followed by postdoc at Harvard Medical School. Then he relocated back to India to start his independent group at THSTI. He has received DBT Welcome India Alliance Intermediate Fellowship, IYBA, and the National Science Bioscience Award. His research interests are in understanding T-cell differentiation pathways in immunity, autoimmunity, and cancer immunity. So looking forward to your talk, Amit.
Thank you very, very much. Thank you. Thank you. Thank you very much for Dr. Rangarajan and Dr. Chandru. It was a great talk, and I'm very honored and humbled to be here and presenting some of the piece of the work what we do in our laboratory. Although my laboratory is kind of very diverse, and the center point of the lab is the so we do very, very diverse work. Is it audible, Piche? Yeah, thank you. So we do a lot of work right from the infectious disease, vaccine development, then immune response to autoimmune diseases. And the key thing is the center point is the immune response. And when we say immune response, again, it's a very complex thing. So we just focus on the T-cell response because we know the T-cells are the long-lasting effector cells, and they convert into memory cells, and that's how we get protection in the vaccine. And these T-cells, once they are dysregulated, can cause autoimmune diseases, and these autoimmune diseases is the chronic inflammation that goes longer. So we'll be focusing today on two things. Basically, I will tell you the three story of the lab, and which will be primarily focusing on the role of interleukin 9. Interleukin 9 is a protein. Somebody who is not from the immunology background, so it's a protein which is produced by the T cells, and this interleukins actually works on various other cell types to bring other immune cells at the site of infection, inflammation, or allergic reaction, and they induces a lot of other reactions. So these cells is very crucial what we have identified in 2008. At the time I was doing postdoc, and this is how we identified this new subset of T cells during my postdoctoral training. And from that time onward, I continuing doing that because it is involved in many, many disease settings, including cancer, infection, as well as allergic inflammation like asthma and allergy. So basically, somebody who is not very familiar about the T-cell response or T-cell differentiation, this is just very, very short, that a naive T-cells, which is normally generated in the thymus, actually comes out of the thymus. They populate the secondary lymphoid organs, spleen, lymph node, and that's where they start searching for antigen. And these antigens are presented by the dendritic cells, which is exposed by, if some pathogens are coming from the blood or under the skin, these dendritic cells actually takes up these antigens, and these dendritic cells move from one place to the other place and goes into the secondary lymphoid organ where these naive T cells are sitting, where they get activated. And the activation of T cells depends on the cytokine in the near milieu, and so therefore the T cells come in different flavors. For example, if you actually have naive T cell which is being exposed to these cytokines, they become Th1 cells, and the Th1 cells are important for elimination of intercellular infection like Mycobacterium tuberculosis is one of the classical examples. So if you have the mutations in these pathways, the Th1 cells cannot be generated, and there are individuals have been identified that they cannot generate Th1 response, and as a result their intercellular infection goes on, and they ultimately lead to death. Similarly, if you have naive T cells exposed to different kind of cytokines, induces Th2 cells, And these Th2 cells are very important for elimination or clearing extracellular pathogens, particularly worm, helminth infection. And if these cells get dysregulated, they causes allergy and asthma. And as you see, there's a lot of pollution lead to the allergy and asthma, and that's what the Th2 cells are important to cause this pathology in the lung. And we have third subset, which is identified in 2006, Th17 cells, and these Th17 cells are involved in any autoimmune disease you name. These are the cells gonna be the culprit. So anytime you have multiple sclerosis or inflammatory bowel disease or any chronic illness, the IL-17 is being produced by these T cells and causing the inflammation. Therefore, you suppress IL-17 using anti-IL-17 antibody, and that's what you get miracle and you get better. So this is a subset which is I'm going to talk about, the Th9 cells, which we have identified, and then we found that these cells are involved primarily in the tumor immune response. On this side, you have these effector cells. On the other side, you have regulatory T cells. They always live in the harmony and the balance. So if you remove these cells from the body, right, there's a way by which you can do in the mice. You create mutant mice, and these mutant mice actually die because these cells will cause havoc and multiple organ failure. The similar kind of setting happens in the human when you have a mutation in the FOXP3 gene, and these humans die very early because of the multiple organ failure of this, these T cells. So this kind of always a balance between effector and regulatory T cells. So I'm going to talk about IL-9. IL-9 initially discovered as a T cell growth factor. This is way back in 1994. And there was less known about the T cell immune response because their technologies were not very, very defined. There was less resolution. So later on, the IL-9 found to be involved in the allergic inflammation, extracellular pathogen elimination, autoimmunity, as well as anti-tumor function. So based on the discovery what we did in 2008, the Th9 cells were involved in various disease settings. You can see here tumor immunity, helminth immunity, inflammatory bowel disease, allergic inflammation, and these are the various publications we have done after identification of these cells in various disease conditions. So one of the interesting things what we really found that after we identified these T cells, a lot of publications came in high impact journals identifying the role of these new subset of T cells which is producing interleukin-9 are involved in the anti-tumor immune response. And you can clearly see that Nature Medicine paper is showing about how these T cells can suppress tumor by various other articles. So then we wanted to really go into the more transcriptional landscape because we wanted to know how these cells are generated. What are the transcriptional factors? What are the various pathways? So therefore, and this is how Th9 cells can actually kill tumor. There's two different kind of mechanism. You have NK cell activation, CD8 response, and on the other hand, you have the mast cells activation, CCL2 mediated pathway, and dendritic cells survival. That's how these cells actually induces its function. So we really wanted to understand how these cells are generated, and therefore we used this strategy. We generated these cells in vitro from the mouse, and then we went for the RNA sequencing to identify what are the different pathways and the transcription factor which is required for these cells. Because if you know these different nodes in the transcriptional regulation, then you can target these cells. Either you can suppress based on the requirement, or you can enhance based on the requirement of the disease conditions. So what we could see, there is a difference. Definitely, there are kind of different sets of genes are being expressed by these cells. And we obviously have heat map, and we found many transcription factors which are kind of up and down in these cells. And we focused on one of the transcription factor called FOXO1, which is here on the screen. And this you can see there's IL-9 and FOXO1. This kind of both are upregulated in the condition where it's supposed to be. So therefore, we focus on the FOXO1 because it's an interesting transcription factor. The role of FOXO1 was not really known in Th9 differentiation, and various other based on the literature, we decided to really focus on this. So what we could first identify what we did. We took this IL-9 promoter or around 2 kb locus, and we scanned this promoter to identify whether this promoter is having FOXO1 binding site or binding motif, because that's what the transcription factor goes and bind and might be transactivating this gene. So this is the first approach we did. And what we could found that within this 2 kb promoter, there was a 4 different FOXO1 binding site what we could identified in this. So that actually indicating us that, yes, there is a possibility and maybe this transcription factor is maybe going to activate this gene. So this is actually we did in the mouse, but then we wanted to make sure what we are doing in the mouse is actually holds true for the human. So therefore what we did another analysis where we did a different conserved region between the mouse, monkey, and human. And we found that this FOXO1 binding motif remained conserved between mouse, monkey, and human at 2 different locus, like CNS2 and conserved CNS0. So it looks like this is going to be same for the human, which we later on concluded. And then we did this chip PCR assay where we pulled out the FOXO1 using magnetic beads, right? And then we just wanted to make sure that whatever the binding motif of the FOXO1 can pull out, therefore there may be a physical interaction of FOXO1 and IL-9 promoter. And what that we could also see by the chip PCR that yes, when you actually pulled out FOXO1, the IL-9 promoter is actually also coming out, and that's what we have done by the sequencing. And ultimately we wanted to make sure that this is really the case, because FOXO1 is important for IL-9 induction in Th9 cells. So therefore we did this experiment where we have deleted FOXO1 using FOXO1 conditional knockout, where we cross this FOXO1 conditional knockout with the CD4 Cree. So in this knockout, the CD4 cells will not have FOXO1, rest of the body cells will have FOXO1. Because FOXO1, if you delete in the whole body system, the mice is actually embryonic lethal, because FOXO1 is important for many other cell types, right? So therefore we did this conditional knockout, and then we found that if you delete FOXO1 in the CD4 cells, you have basically ablate totally IL-9 as compared to control, and that's what we also see by the real-time PCR as well as the FACS. So ultimately we wanted to take this to the next level, so we have to show this in the disease setting. So what we have done allergy-induced mouse model where we have actually took biopsy mice. We actually sensitized them with the OVA, with the intraperitoneal injection with the OVA with the alum, and then later on we came back with the intranasal dose of this OVA peptide, so that these mice can actually induce a similar kind of this breathing problem, and which we measure as a PANH index. So what we did, we actually silenced the FOXO1 using FOXO1 siRNA in this case, and what you can clearly see that when you actually block FOXO-Y siRNA, you see a PANH index is actually substantially less in this case. We collected the lung lavage from these mice to measure the interleukin-9 in the lavage, and clearly see when you actually block FOXO1, the level of IL-9 actually decreased significantly. And IL-9 is one of the factors which is important for mucus secretion in the lung. So if you have more mucus in the lung, that means you have a breathing issue, right? So therefore, in case of FOXO siRNA knockdown setting, you have less this mucus producing cell we call PAS staining is lesser in case of this. So then we actually make a conclusion that that FOXO1 is a transcription factor important for Th9 cells, and it binds to both IL-9 and human gene locus, and it actually abrogation or deletion abrogates Th9 cell differentiation. So then we continued this role of IL-9 and transcription regulation to go into more details and what are the other transcriptional landscape or the factors. So one of the things from our RNA-seq we analyzed, and one of the pathway which we found highly enriched in the Th9 cells is the EGFR pathway. And then we did confirm this RNA-seq data using differentiation, and we found that EGFR, one of the receptor, is highly expressed on these cells. And when we again did a conditional knockout where we deleted EGFR conditionally in the CD4 cells, using conditional knockout, we see this lesser IL-9 being produced by the Th9 cells if you delete this. So EGFR is actually a molecule which is normally being targeted in the tumor, and then there is a different molecule by which a small molecule you can block EGFR. And one of the molecule is called, a gefitinib is a blocker of EGFR. So what we did, we use a model where we have melanoma cells expressing OVA, right? And then we took the OT2 transgenic Th9 cells, and then we treat these cells with the EGFR inhibitor and transplant these cells in the mouse. And we clearly see that if you block, gefitinib as a EGFR, you can see Th9 cells which otherwise inducing anti-tumor function, which is in the green. And if you block EGFR by gefitinib, you lose the anti-tumor function of Th9 cells in this case. So EGFR is actually having 7 different ligands, and that depends on where EGFR is being expressed, right? So therefore, we wanted to focus which ligand in this case is playing a role in the model. And we focused on the amphiregulin, which is one of the ligand is being produced by the immune cells. And there are a couple of reports is indicating that amphiregulin is produced by the T cells, and there is an important role in T cells. So therefore, we focused on the amphiregulin. And we asked the question, maybe the amphiregulin is maybe being produced by the T cells, and that may be feeding back to these Th9 cells. So therefore, we did some experiment, and what we found, obviously, in the Th9 cells, you have more amphiregulin, right? And if you actually block amphiregulin by anti-amphiregulin antibodies, there's a less Th9 differentiation. And actually, this we did confound by genetically using amphiregulin knockout mouse. So what we have done, we have taken the wild-type Th9 cells and amphiregulin Th9 cells, and we did RNA sequencing. And what we found, there's a kind of lot of other genes are up and down, but obviously IL-9, which is one of our signature genes, is actually down in case of amphiregulin knockout mouse, indicating that this is the pathway which is operating in Th9 cells. So EGFR is known to actually lead to the activation of HIF1 alpha. This is one of the major node the EGFR lead to in case of cancer settings. So what we found in our RNA-seq that response to hypoxia and glycolytic process is kind of upregulated. And then we actually, glycolysis again lead to this hypoxia, and hypoxia is a HIF1 alpha is a major transcription factor. So what we found that, HIF1 alpha is also involved in the Th9 differentiation, both by luciferase we confirmed, and then subsequently we blocked the, HIF1 alpha by acriflavin, one of the inhibitor. We found that Th9 function is actually substantially reduced in case of tumor immune response. HIF1 alpha is somehow involved in many other, metabolic regulations. So we did the detailed metabolomics using the wild type and HIF1 alpha knockout mice, and we focus on one of the factor which is alpha ketoglutarate, which ultimately lead to succinate if you go to the Krebs cycle. And succinate is one of the metabolite which actually stabilize HIF1 alpha. So what we found that if you have succinate, your HIF1 alpha and IL-9 induction is further up. And then when we actually treat these cells on the mice with the succinate, you see lesser tumor. So this is what we could actually show and publish, so that we have these two cytokines induces EGFR, and this EGFR bind to amphiregulin, and amphiregulin ultimately lead to HIF1 alpha, and that HIF1 alpha bind to and transactivate IL-9 gene, and that produces more IL-9, and that induces tumor killing. So far so good, and actually we took this advantage of having these Th9 cells in the COVID-19 because we know that IL-9 is important in the lung inflammation, and that is in allergy, and IL-9 receptor and IL-9 gene both are genetically linked to asthma. So we thought maybe in the COVID-19, because it's a lung disease and there's a lot of mucus we see in the COVID-19, even in the mice, hamster, and the human. So we took this advantage, and since we have this facility where we were doing COVID, 5 minutes. So when you can see the cytokine storm, you have dexamethasone, which is known to suppress cytokine storm. And then what we could really see, the lot of mast cells is accumulated in the COVID-19, and IL-9 is one of the growth factor for mast cells. So we got an idea that maybe IL-9 is doing a lot of things in the COVID-19. And what we clearly see in case of human, if you take active COVID-19 disease samples, You see a lot of IL-9 in the COVID-19 as compared to healthy. We replicate the same thing in the hamster model where we put active COVID-19 virus. You see more IL-9. We did in the mouse, so human, hamster, mice, in 3 conditions, we have a lot of IL-9. And then what we did, first thing, to neutralize IL-9 using anti-IL-9 antibody. And we took remdesivir as a positive control, which is an anti-COVID-19 drug at that time, because there was nothing was available. And we clearly see that this, anti-IL-9, you can see green one, is much better even the remdesivir. It's actually the weight loss is not really significantly reduced when you actually neutralize IL-9. And you can clearly see viral load is also less when you neutralize IL-9. And the viral, N protein is also much, much lesser in the treatment group. So therefore, we found that, when you have anti-IL-9 treatment, there's a lot of antiviral genes are upregulated, and that may be causing the, virus to control. And then we did, combination treatment with anti-IL-9 antibody and the remdesivir, and we found when we combined both of them, lower dose, we found a better effect. So it's kind of therapy we wanted to, say. I'll just quickly skip this. So one of the interesting thing what we found in our what we published FOXO1. So we had this FOXO1 knockout mice, and this FOXO1 knockout mice, we crossed it with the ACE2 transgenic mouse just to see what happens to the phenotype. And we could clearly see here one of the interesting finding here. If you can just click on this video on this. Yeah. So these are the mice. Normally when you infect with the COVID, they just knocked out in 4-5 days. They can't move, and they ultimately lead to that in 7 days. And if you see this is a wild type mice, and when we actually cross this with the FOXO knockout, they are all healthy. They are just like, healthy mice. There is nothing happening to them. So it looks like, FOXO1 and IL-9 axis is somehow managing these disease not to happen. So therefore, what we proposed, and we did an experiment, that if you use FOXO inhibitor, and you actually modify this drug, you can use this as a drug. So based on this, we found, so now we can go to next slide, yeah. So based on this, we can clearly see that, this inhibitor can be very much useful. And there are a lot of other experiments we did, but I want to just come to a conclusion. This is another one. So if you take the FOXO knockout, when you actually put recombinant IL-9 into them through the intranasal route, they're again having disease. So ultimately, what we could see here in the COVID-19, there's a lot of IL-9 is being produced, and this IL-9 is actually regulated by a FOXO1. And if you somehow treat these mice with the FOXO1 inhibitor or IL-9, ultimately you have less pathology, less viral infection, because you have more antiviral pathways are upregulated. And these are the various funding agencies actually supported us, and these are the warrior of the lab, because our lab was 24/7 was open in COVID-19, and thanks to CDRI at that time, because most of the hamster we were actually importing from you for doing all these work. So thank you very, very much, and I will be happy to answer any question regarding this or anything with what my lab actually does. Thank you so much.
Thank you, Amit, for sharing. Time for one quick question. Yes, Monica.
Neutralized, but the balance is important. Did you give further infection, any other infection or tumor progression? I mean, how exactly their immune system will behave?
Yeah, we know. So basically these knockout, when we generate, although they are conditional knockout, but if you keep them for a longer time, so we made a cohort of these mice and we kept longer time for a year, right? Normally they are 2-year mice, they survive 2 years. They generate autoimmune disease by itself, and particularly the disease in the brain. So they have paralysis, and they have kind of multiple sclerosis kind of disease. So because your immune system is kind of activated if you don't have FOXO1, it's a kind of keeping the balance. If you remove the brake, the T cells start infiltrating in the brain, and as a result, these mice actually having a paralysis, like very similar multiple sclerosis. Yeah. Yeah, so basically in case of, if you have this inhibitor, which is, genetically obviously we cannot target because it's a very critical balance, but if you have this inhibitor which is on and off, I think can be beneficial for conditions when we have conditions like COVID-19, when we have no idea people were dying. I think that time you have to just give this inhibitor and people will be saved. Yeah.
I see a burning question from Manoj. This is the last question.
So, very nice work. Congratulations for that. A quick question. Can you comment on the proteins which are regulating HIF1 alpha and FOXO1 in your experiments?
The proteins which are regulating both of
FOXO1,
these.
because you showed all the effects downstream of FOXO1 and HIF1
Right.
alpha. I was just
Yeah,
wondering.
so I got your question. So basically, this is all in the paper. Because of the time, I did not. So the FOXO1 is actually regulated from the upstream. It's a kinase called AKT, PI3 kinase AKT pathway. So first is the receptor. The receptor transmits the signal to PI3 kinase and AKT, and this phosphorylated AKT phosphorylate FOXO1. So FOXO1 normally remain in the nucleus. Once it is phosphorylated at 3 different sites, it goes out of the nucleus, loses its transcriptional activity, and get degraded by ubiquitin. Yeah, so FOXO1 can be regulated by phosphorylation, acetylation also. In other setting, it has been shown. So there's a 2-3 different pathway by which the FOXO1 can be regulated upstream than AKT, one of the node, which we have shown in the paper, AKT. We use AKT dominant negative, AKT dominant active, inhibitor, various experiments we have done in NatureCom paper. Yeah.
Okay. Great. Thank you so much, Amit, for
Thank you.
sharing such exciting stories. Since Amit has to leave, so we'll have the felicitation for him right now. I request the director and Professor Vijay Chandru to please come up on the stage. Great work.
Thank you.
A lot of questions, but I think email has to be written to you. For that.
Thank you. I'm very sorry I had to leave because I had to
Vijay, why don't you do the
Thank you very much. I'm very honored.
come to the side?
Sure.
Okay, so now we move on to the presentation from chemical sciences, and we have Dr. Ramasastry from Aizer Mohali. He does his PhD from ISC Bangalore, followed by a postdoc at Scripps Institute. He also has an industry stint. He has worked at Jubilant and Syngene before joining Aizer Mohali. He is the recipient of bronze medal by CSIR and also has Swan Jayanti Fellowship. His research work focuses on design of synthetic routes to incorporate asymmetric synthesis, diversity-oriented approaches for green and sustainable chemistry. And his group has also tried one-pot catalysis. And over to Ram for his presentation.
Okay, thank you very much, organizers, especially CDRI for the invitation and also wonderful hospitality. Great to be here. It's my first visit to CDRI and feels wonderful to be among some of the great scientists sitting in front of me. Today, I'm going to talk about some of our work as titled Palladium Catalyzed Cycloisomerization Reactions. At this time, let me also once again thank the organizers for having my talk after Professor Visai's talk. He actually provided the necessary introduction or the background to my talk. He actually pitched my talk in an appropriate manner. He rightly pointed out the significance of natural products in drug discovery And he also, of course, showcased their own efforts leading to synthesis of several complex natural products. Our group at IISER Mohali works on these areas, catalysis by transition metals. Complementary to metal catalysis also we work on, that is phosphine chemistry, that is organocatalysis, and also recently we got into sulfur-related chemistry. We primarily aim to develop new methods, but our always intention is to extend the scope of the work to synthesis of especially bioactive natural products. In fact, in this talk I'm going to showcase how we developed a concept-based approach and extended to synthesizing some relevant natural products, not as complex as what Professor Bisai has shown. As of palladium catalyzed coupling reactions, the audience must be aware of especially the Nobel Prize winning reactions such as Heck, Suzuki, Negishi coupling reactions. Others also, of course, are quite popular as of making carbon-carbon as well as carbon heteroatom bond reactions. Among them, our group especially focuses on Suzy-Trost chemistry, which is otherwise known as allylic alkylation chemistry. But our group especially employs palladium as the medium. Unlike many other metals, the reaction which is nowadays known. My talk, I can tell you, if you have basic understanding in chemistry, I'm very sure that it is at a very fundamental level. Any of you can follow with basic understanding, and in fact, I'm going to give you, if you can understand this reaction, rest of it is quite straightforward. That is what I titled as Suzy-Trost reaction. This is effectively the allylic alkylation reaction, as I told before, involves the replacement or substitution of a leaving group in case of allylic substrates of this kind in presence of palladium, which forms a pi-allyl palladium species of this kind. And appropriate nucleophile now traps this electrophilic palladium species, forming two types of products. This was demonstrated first by Suzy and then made extraordinary efforts by Trost and co-workers. Indeed, the name comes with their names appropriately. One day, of course, this is how my teaching helped me. I should tell you the basis for what I'm going to talk to you is entirely based on the kind of teaching that I have done. One day I came out of my class and thought, okay, this is known with allylic substrates. Why not, or what about dianylic substrates? So of this kind. If you have dianylic substrates, two types of substrates are possible, and in presence of a palladium catalyst, it can form two types of vinyl pi-allyl palladium species, as you can understand. For example, this substrate can form this kind of pi-allyl palladium species in analogy to what you see at the top, and this substrate can form, of course, this or the other type of pi-allyl palladium species. What is interesting to realize here, unlike two products that can be obtained in this system, you can actually obtain three products, at least theoretically, as you can see, right? One where nucleophile adds at this position, one here, one here. This was demonstrated, although not extensively, but Trost already made reasonable efforts as of this particular chemistry is concerned. What especially attracted our attention is the intermediate that is highlighted here. If you see carefully, as I told earlier, the electrophilic pi-allyl palladium is at one end, and as you can see, sort of electron-rich system in the sense in the form of the pi-electron cloud on the other end. We thought we have now electrophilic system on one end, sort of nucleophilic system on the other end, depending on, of course, what is the R group there. If we bring them together somehow, it's like, if you imagine a stick and if I somehow bend it so that the two ends come together, can I now make a ring out of it? Okay, that was the idea, rather than what was known earlier. Indeed, accordingly, what we have done, we just made or utilized a simple trick. We just introduced substituents such as R2 and R3 onto this same dianylic substrate that you saw a little while ago. What we believe is that these R2, R3 groups possibly exert so-called 1,3 axial strain, which is similar to cyclohexanes that we talk of, the axial interactions. And that may facilitate reorganizing this dianylic substrate to a form such as this. Now that can potentially bring together these two reactive ends highlighted in blue, so that an E-type reaction potentially possible. And that leads to the formation of this allylic cationic system. Eventual proton loss can lead to cyclopentadienes of this kind. That is a simple proposition. Indeed, it worked in our favor when we actually replaced methyl groups, just methyl groups in place of this R2 and R3 that I showed in the last slide. It actually gives the product expected, but of course, this is not the same what we've thought. Indeed, 3A and 4A were the ones actually originally hypothesized products, but we actually got the other possible isomer, that is this one, which can be easily distinguished, as you can see, from the rest of the two, just based on proton NMR, just that this CH2 is present, which is absent in these two. With this technique, we could able to make a variety of tetra-substituted cyclopentadienes. Needless to say, to this August gathering, the significance of cyclopentadienes, not just in Diels-Alder chemistry, even in forming metal complexes, for example, they can find excellent applications. Eventually, what we have also done is to introduce fusion across one of the double bonds that you saw. Earlier, it was linear dianylic acetate. Now we introduce fusion in the form of arene or a heteroarene appropriate. Thereby, you can actually access appropriate fused cyclopentadienes such as indenes or cyclopentene-fused arenes and heteroarenes, as you see here. So we didn't stop there. Of course, I'm actually cutting short some of other works done as part of this study. We eventually extended the scope of this work to synthesizing natural products. We found that that these molecules, these bioactive natural products, these are the molecules belonging to resveratrol family of natural products. We thought that passifloral F is suitable for this synthesis. Applying this strategy. For that purpose, we made this appropriate acetate starting from traditional synthetic chemistry, and that was subjected to the optimized conditions that are developed by us that provided access to these indenes. This indene was hydrogenated followed by selenium oxidation. Our intention was to achieve actually benzylic oxidation, but it over-oxidized to give this enone or otherwise indenone. This was shown earlier by Sarpong and co-workers in synthesizing passifloral F. So it constitutes a formal total synthesis that way. So what you have seen in the last couple of slides is an isonazaro type cyclization, if you can envision. It involves basically substrate of this kind in presence of palladium. You can see the straightforward nature of the reaction conditions as well, gave access to cyclopentadienes. So we questioned, can we extend this strategy to Nazaro type cyclization? It only requires manipulation of the substrate. You can see that this kind of dianylic acetate is manipulated to this diacetate, this dianylic acetate. We believe that in presence of palladium, it can potentially form this parallel palladium species. Now, this arene or heteroarene can offer ene-type cyclization, or of course, different notation or different naming also you can give, such as Fidelcraft's type alkylation, but providing access to cyclopentene-fused arenes or heteroarenes. Unlike the earlier strategy, this gives us opportunity to introduce a quaternary carbon or a spirocarbon, depending on the nature of that R1 and R2. Right. So in this context, let me highlight what are the Nazaro type cyclizations known utilizing palladium. Usually, as you all know, Nazaro reaction is something that is traditionally facilitated by acids. But there are some acid-free methods for Nazaro cyclization, as you see here. Only 2 reports, at least to my knowledge. That is, this dienone in presence of palladium gives access to this cyclopentenone, alpha hydroxy cyclopentenone that was developed by TS. This is otherwise famously called TS Nazaro reaction. On the other hand, Frontier also developed the cyclization in presence of these cationic palladium-2 complexes. So, but you can see the difference between the substrate design. This is a dienone versus the dianylic, sort of dianylic acetate that is what we hypothesized. Indeed, what happened, as you can see, after several efforts, the substrate design that you're seeing here, it didn't come overnight, obviously. After several designs, we realized that substrate of this kind, which is effectively a dianylic acetate system, in presence of simple palladium acetate, palladium chloride, as you see here, gives access to these spiroxindole systems. You can see a variety of heteroarene and arene-fused spiroxindoles can be assembled, not just from C2 position, even from C3 position. This is equally feasible. That also gave us initial indication that it may not be proceeding through CH activation process. It is most likely going through the kind of hypothesized intermediate, such as this parallel palladium species. But during the substrate scope evaluation, we made an interesting observation. Please note this one, that as long as this reaction works, as long as this R1 is a substituent. Okay, that R1 is nothing but in the form of this methyl group or benzyl or whatsoever in these examples. Once this R1 is a H, I mean unsubstituted, the reaction doesn't give the expected product. It only gives deacetyloxylated product, which is not unexpected under palladium conditions. So at this stage, of course, we didn't realize. We thought initially it's something problem from our side, but we went ahead to even extend this scope of this work in creating a quaternary center, all carbon quaternary site. You can see natural product like structures, tricycles, tetracycles, even pentacycles such as this can be assembled quite easily. It's only about how you or what kind of substrate or what kind of starting compound you synthesize. The respective compound can be obtained in healthy yields, as you see across the examples. These are only partial list of examples that I'm depicting among the ones that we've actually made. And not just indines, you can also see that cyclopentene-fused benzothiophenes, indoles can also be made using this protocol. Here also, we saw interestingly that whenever the substituent is missing in place of this R1, which is next to that acetate, you can remember, the reaction didn't work as per our expectation. In this case, especially, it gave complex mixture. We'll come to that during describing the mechanism. But otherwise, we also extended this work to synthesizing natural product. For example, we identified several natural products in this core. For your information, nature produces several cyclopentene-fused indoles as the primary component in it. Among them, of course, I'm listing only few of them, out of which we actually prepared thionequinone H and dicroinone. In fact, Professor Bisai also prepared at one stage of his career these molecules. For this purpose, of course, he used acid medium to accomplish this transformation. What we've done, we prepared this dianylic acetate, otherwise benzylic allylic acetate, using standard synthetic protocols, and then subjected to the optimized conditions, gave access to this indene system. And that was subjected to CAN oxidation, that is effectively deprotecting the methoxy groups, and that gave access to thionequinone H, the natural product. Eventual demethylation using basic conditions gave access to another natural product, that is dicroinone. This is what, as of establishing the concept versus application and the demonstration. Now coming to the mechanism, after a lot of efforts, of course, the mechanism was not accepted as you see on the screen in the first attempt. We had to make a lot of changes and a lot of efforts, generating evidence in favor of this particular mechanism. So we believe otherwise that palladium forms the parallel palladium species, that is what is initially of this kind. This, we believe, reorganizes owing to the steric considerations to this particular system. As you see that when it reorganized, the two ends, the reactive ends come together. That is this end, that is the reactive site, and this end come together. That is what is this particular species. This now undergoes N-type cyclization. Otherwise, you can think of as a Friedel-Crafts type cyclization, giving access to this particular species, and eventually the elimination of palladium regeneration gives access to these compounds that you saw so far. Okay, as you can see here, we also resorted to kinetic isotope effect, which gave point for ruling out the CH activation as of this particular cyclization is concerned. Okay, we also extended this concept to creating tertiary center. Of course, a relatively easier challenge, as you can understand, that gave several types of indines. Now with monosubstituents, as you can see, unlike disubstituents, which were what we saw as quaternary or spirocenters, cyclopentene-fused indoles, benzothiophenes fused to cyclopentenes. Okay, and here also we made complex analogs of some natural products. As you see, pentacyclic structures can be obtained via mere manipulation of the starting compound. These compounds can be obtained, let me tell you, in 2 steps, if at all you can organize the respective fragments. And these molecules, as you can see, are the part structures of these natural products, are the BMS compound that is here, On the other hand, we also showed the application in generating these pentacycles, such as what you're seeing here, which find important medicinal material applications. We also demonstrated the scope of this work in establishing one-port double annulation. Of course, just so far what you have seen, one annulation or one cyclization. Now we have performed two cyclizations simultaneously. But more importantly, it's not about two cyclizations. It is about creating indocines which otherwise require multi-step synthesis. They are known to exhibit interesting physicochemical properties. We didn't stop there. We also established the scope of this work in synthesizing the beta diacerone. As you see here, this is non-natural analog of the natural product. The natural product is gamma diacerone, but this is reported to be a potential natural product that may be isolated or so. But that is what is the beta diacerone. As you can see, the difference is that these all syn arms that this Indian moiety has. For that purpose, we prepared the appropriate allyl pivalate. Somehow we couldn't make the acetate in this case, but we made the pivalate, and that pivalate responds equally efficiently under these conditions, as you can see, in generating this indene. Subsequent transformations were rather straightforward: hydrogenation, selenium oxidation, and once this enone was obtained, that was subjected to ethyl magnesium reaction, followed by selective dehydroxylation, giving access to this diene moiety, which eventually was hydrogenated to give selectively the beta diacerone. We were fortunate that we could able to get the crystal structure. In addition, before we got the crystal structure, we could able to establish this in selectivity by extensive 2D data. So far what you have seen was our efforts with monoacetates, otherwise monoallylic acetates. We also thought what can happen with allylic diacetates, which are very less explored, unlike allyl acetates or allylic activated substrates. Trost didn't leave this area as well. Trost earlier established that diacetates, allylic diacetates of this kind, can be considered equivalents of 1,1 dicationic equivalents or 1,3 dicationic equivalents. What that means is he showed through these studies that you see here that the allylic diacetates, for example here, can be considered equivalents of 1,1 dicationic equivalents through the formation of these cyclopropanes, as you see here, using the dianionic systems that he employed here. And he also showed the 1,3 dicationic equivalency through these examples. As you see here, the allylic diacetate that you took initially was reacted with one nucleophile, you got allylic acetate. Again, he performed another reaction stepwise to introduce another nucleophile, effectively showcasing 1,3 dianionic equivalency. Now what you thought at this point, can we now but what to notice as of this particular study is this is stepwise extension. But what you thought, can we do this in a cascade manner? Can we trap this allylic diacetate in an effort leading to a cyclic structure, which is otherwise what we call annulation? So for this purpose, we considered the phenols or naphthols as 1,3 dianionic equivalents. And of course, the 1,3 dicationic equivalence was already demonstrated by Tross. But what is interesting here is that if at all the concept works, it leads to annulation leading to chromine moieties, if at all the case. So I'll skip this portion, but otherwise this is the result. Indeed, we were successful actually achieving this. The allylic diacetates were shown to be very good substrates as of reactants as of their reaction with naphthals or otherwise meta-substituted phenols. Especially, you see several naphthals we employed, but not all phenols worked. Surprisingly, only meta-substituted phenols worked. Please note this point. I'll come to this a little later. But you can see the scope of this work. You can generate variety of chromines, even spirochromines. We have indeed shown photochromic applications of these molecules, which I'm going to show very briefly in one slide. Nevertheless, we showed the scope of this work in synthesizing some natural products. As you see here, the method was applied on this hydroxy carbazole. As you see, treated with this allylic diacetate, we got access to this, which was eventually extended to synthesizing these natural products, grenambine, methionine, muramycin natural products. This also establishes formal synthesis of the natural products listed there. We didn't stop there. We also extended the scope of this work to the shorter synthesis of bohenol D. For this purpose, we made this meta-substituted phenol, and that was treated with this allylic diacetate, gave access to the natural product. I showed you, of course, this is the mechanism. I told you that meta-substituted phenols only work. Why? That is what we believe, that after the initial parallel palladium complex formation, initially the naphthols or meta-substituted phenols react. We believe that the meta-substituent here exerts sort of steric encumbrance as of these substituents are concerned. So you can see that 4 potential intermediates are possible. Among them, due to steric reasons, we believe that the substrate itself reorganizes to something of this kind. You can see how it goes because of paucity of time. I'm not getting into the details, but this eventually undergoes reductive alkylation, and subsequent elimination of palladium or regeneration of palladium gives access to chromines that you see there. Okay, that's how the meta-substituted phenol, meta-substituent, we believe plays the key role. This is my last slide. We also showed the photochromic applications of these molecules. You can see this was done in collaboration with my colleague, Professor Sukumar Venkatramani. He has shown not only in the liquid phase, even in solid phase photochromism, we could able to successfully demonstrate. With that, I'll almost conclude my technical aspect. Our efforts in this area, the metal catalyzed transformations were invited through various reviews, as you see here. And then, as I mentioned in the beginning, not just the metal chemistry, we also pursue complementary chemistry, metal-free. One among them is phosphine catalysis. These are some of the summarized contributions, not all, of course. We worked in the development of NH-selective Morita-Bellis-Tillman chemistry. We also used phosphines in the sort of first time demonstration of intramolecular hydroalkylation chemistry, intramolecular hydroacylation chemistry. We also demonstrated the scope in the reductive aldol chemistry, vinylogous aldol chemistry, and some others which are not listed here. We've been accidentally got into the sulfurylate chemistry. Sulfurylates typically, which are known to the chemists as Koryakovsky reagent, for example. We use Koryakovsky reagent not for cyclopropenations. Okay, you can refer to this chemistry. It leads to cascade transformations, providing access to unprecedented scaffolds. Okay, that probably I'll talk to you when I visit next time to CDRI. With this, let me thank my colleagues. Because of their contribution, I'm here, obviously. The ones especially highlighted in red are the contributors for the talk today. This is my, not current group, of course, a little old. Some people left, some people joined. After this, let me thank the funding agencies and CDRI again for recognizing our efforts. All of you for your great attention. Thank you very much.
Thank you, Ram, for giving such a lucid presentation and giving an overview of different projects going in the lab. We have time for one quick question. Oh, 2 are there. Okay, so I give chance to both of you. Ravinder.
So, needless to say, I think very nice strategy. My curiosity actually on Nazarov type of cyclization reactions. So curiosity is that what happens with the electron deficient arenes for this cyclization. Another one is the residual selectivity when this arene group is substituted.
Yeah, so obviously electron withdrawing group don't favor the reaction. That is one. Second is because these are intramolecular, there are hardly any chance for any other regioisomers that one can think of. If it is intramolecular, most likely there can be a chance of side products, I mean unexpected reactions or due to regioisomers or whatsoever. This is, most of these reactions you would have seen, common thing among all of them is intramolecular. They're more or less sort of defined as of substituents versus geometries, and also sort of defined to get only one product, mostly one product. So we didn't observe any other products seriously. I mean, that is the answer to your question.
Okay, Nilanjana.
Thank you for such an excellent talk. So So, in the first work, you have shown the synthesis of cyclopentadiene.
Right.
So, did you by any chance see any possibility of dimer formation?
No.
Is it because of the high substitution
Exactly.
in the substrate?
Yeah, yeah.
Okay.
That is the answer. Yeah.
Okay.
So these are because highly substituted, not immediately dimerizable. But you can actually that's why I said at that point, if you remember, several details I skipped owing to the interest of time. We actually demonstrated Diels-Alder chemistry and presented those details also. But that requires forcing conditions. A typical cyclopentadienes dimerize even below room temperature. But these require some of forcing condition because of substituents.
And some of your works have actually very good opportunity of asymmetric approaches.
Right.
So did you?
Yeah, we tried, but we didn't get the required enantiomer selectivity for publication purposes.
Okay.
We get up to around 60% or so, and we didn't proceed to publish. But we mentioned that in the papers.
Okay.
That these are the preliminary results leading to enantiomer selective synthesis. But we didn't develop that as an enantiomer selective version, because obviously 60 is not nowadays acceptable.
Oh.
Yeah. Yeah, that is known already. That is what Bisai and several others have done. That is known. With alcohols, it's known. Our intention was to develop acid-free version. That's what you see across. OBOC also works. OBOC, OSC. OMS, ODS we didn't try, but probably they may work, but we didn't try. Yeah, but difficulty in synthesizing also several times, because for students, acetate making is like just in a simple step for them. Yeah.
Okay, great. Thank you, Ram. And now we move on to the last speaker, Dr. M.S. Reddy. Oh, yeah. Okay. So he did his, So Dr. M.S. Reddy has received CDRI award in biological sciences category. He received his PhD from University of Manitoba, Canada, and conducted his postdoctoral work at Yale University. He relocated back to India and started his independent group at CDFT. He's recipient of Wellcome Trust Intermediate and Senior Fellowships, and his research group is trying to decipher molecular mechanism of various cellular signaling pathways and their contribution to human diseases. So we look forward to your talk, Madhika.
Thank you. Am I audible? Yeah. Thank you, CDRI, for having me here. So for next 20 minutes, I'll try to give a glimpse of what we have been doing in the lab for the last 13, 14 years. It's a challenge to complete in 20 minutes, but I'll try. So what we are interested in the lab is looking at the systems that regulate, that are important for maintaining cellular homeostasis. And the two systems that we are broadly interested are in phosphatase biology and the ubiquitin biology. While many of you probably know, ubiquitin is this set of protein is important for maintaining homeostasis at the protein level by degradation or any other mechanisms. Phosphatase are the ones which maintain homeostasis at the phosphoprotein level. So I'm not going to talk about ubiquitin today because of the time, so I'll talk only about the phosphatase. All the work in the lab is supported by India Alliance and other funding agencies, before I forget. Thank you. So to start with, I think it's a simple, straightforward slide where you can see proteins get phosphorylated at 3 different residues, and there are more residues now, through set of enzymes called protein kinases. And this is not a unidirectional mechanism, so this needs to be removed by a set of enzymes called phosphatases. This is a very simple basic slide, high school slide. What people are interested in the field is all focused on protein kinases because of their druggability or whatever reasons, right? Easy to study. What are phosphatases doing in the field is they're forgotten. So not many people are interested for many reasons. We are one of those labs which are who are interested in studying phosphatases. And if you look at human phosphatases, there are around 210 of them.
classified into different classes based on their domain architecture and the kind of substrates they act on. So you can see serine, threonine, phosphatases, tyrosine, and there are phosphatases which work on both residues. There are histidine phosphatases, and there is a separate class called lipid phosphatases as well. So I'm going to talk about only the protein phosphatases today. And these are also again classified based on their cellular localization, either receptor type or non-receptor type, or metal dependent or metal independent. So there are different classes. So the questions that we are asking in this project, in this theme, is three broad questions. So one of them is, what is the functional rapporteur of all these phosphatases in humans? How are limited number of these enzymes? As I said, there are 210 of them. Out of that, 160 are protein phosphatases, right? So how are these limited number of enzymes, 160 of them, able to control all the phosphorylation dynamics of the cell? If you imagine a cell proteome, so it codes around 25,000 genes, but at the proteome level, if you consider all the isoforms, splice variants, everything, it comes to 90,000 odd proteins. So you have 160 proteins, and every protein in the cell, almost 90,000 proteins get phosphorylated. So how is this 160 set of enzymes are able to control the phosphorylation? So that's one another question. And the other point that we are trying to address is, can new mechanisms be attributed to known phosphatase functions for this whatever known already? So these are some of the examples that we came up with. These are some of the stories that we ended up putting together. I'm not going to talk about all of them. The questions that we are addressing here is done through a very simple proteomic approach. So the approach is described here. So what we started, when we started the lab, what we envisaged is to build a protein interaction network of all the human phosphatases, right? So the approach is simple. So we try to put each individual phosphatase into this vector and then make stable line of that, and do a simple proteome, simple two-step affinity chromatography purification, and then elute, do a mass spec, lot of mass spec analysis through LCM SMS, and then do a lot of data crunching to generate a phosphatase interacting network of all the human phosphatases, right? So after doing several years of this proteomic work, so we ended up with this network. You don't need to understand what these dots are. What you should appreciate is, this is the human interaction network of all the phosphatases. So the center ones are the number of phosphatases, and the ones which are blue coded here are the interacting proteins. So if you look at the numbers, so we were able to purify the complexes of 135 of them so far, which gave us nearly 70,000 interactions, and putting a lot of cutoffs. So we could get around 6,000 high confident interactions among 2,000 high confident interaction proteins. What is very interesting is that there are almost 1,000 of them are unique interactions, right? So when I say unique, when one interactor is binding to one phosphatase that is not found in another phosphatase. So these are, there are around 1,000 of them. So essentially you can have 1,000 PhD students working on this, but yeah. So we cannot afford that, but we had to start somewhere. So what is also important is that there are 90% of these phosphatases are unknown in the literature, right? So we could get 10% of these known interactions, 90% is unknown. So it's a rich resource that we have in the lab right now. So to address each of these three questions, I'll put three examples. So the first question is going back, can we fill, using this interactome data, can we fill the mechanistic links, right? So I'll use a very simple, our favorite phosphatase to start with. So it's a phosphatase called P10. It's a tumor suppressor phosphatase. It's the second most mutated gene in human cancers after P53. It's a very interesting phosphatase in the sense that it is a lipid phosphatase to start with, but it is also known to be a protein phosphatase now, right? So what it does is, in a simple term, it is a negative regulator of PI3 kinase AKT growth factor signaling pathway, right? So what it does is, this P10 is a phosphatase which dephosphorylates the PIP3, which is a lipid, back to PIP2. So the PIP3 is required for the activation of AKT, which is initiated by the growth factors, right? So when you have PIP3 conversion, AKT gets activated and then can act on multiple substrates in the cell. But P10 basically negatively regulates this by converting this back. So it is very well studied. Hundreds of labs are working on this.
What is interesting is, given that it is a highly mutated gene, it's not surprising that it has to be regulated in multiple mechanisms. So one of the mechanisms that has been proposed for its regulation was done by Pierre Pandolfi's group, where they showed that ubiquitination, which is shown to be a mechanism for regulating P10, they showed that P10 ubiquitination is important for regulating its localization as well as degradation, right? So this was done in 2008. Until then, P10 was not known to be ubiquitinated. So they also found E3 ubiquitin ligase, which is an enzyme which transfers the ubiquitin onto this substrate, as NET4-1. So this is the E3 ligase that is shown to be ligase for P10, right? So all well and good in 2008, but the twist in the story came when Daniel Oretin's group published a paper where they made a knockout of NET4, and they showed that it is completely dispensable for P10 regulation, right? So NET4 does not do anything with P10, right? It does not change the stability. It does neither changes nor changes the localization. So this is where the field wasn't fixed. So we had to come and address this. So we took the challenge and then tried to. This was the time when we had this P10 interaction map. So that was the first year of my establishment of the lab in CDFD. So we had this P10 interaction map, and we went on search specifically for the ones which are known to be E3 ubiquitin ligases, right? So this turned out to be the E3 ubiquitin ligase that is belonging to the same family as NET4, but it is a different E3 ligase. So what we went on to show is that it is not the NET4 which is regulating P10, which is binding P10. It's the WWP2, which is the E3 ligase, new E3 ligase, and that is the most well accepted E3 ligase. That is the only accepted E3 ligase so far in the field for P10 regulation. We showed that it is specific in ubiquitinating P10, and not only that, it is physiologically and pathologically relevant. So when you when you make this WP2 shRNA cells, they are not able to form tumors. It's very simple. WP2 is able to regulate P10 levels. So P10 levels stability is tumor suppressor is down. So they no longer form tumors, right? So that's first question, answer for the first question. So can we address new mechanisms? This is one of the new mechanisms for P10 that we could come up with interactome data. So the second question is a little more broader, where we wanted to see how are these limited number of phosphatases control the whole human proteome. So the answer for that lies in the way that the phosphatases are not alone, right? They diversify their functions by having multimeric complexes assembled. So if you think C is the enzyme here, catalytic subunit of a phosphatase. So there are different sets of regulators like A and B regulators, which can form holoenzymes and then give diversity to the substrates in the cell. So, but the catch is that among all these phosphatases, only 7 of them, which are belonging to this PPP family, right, are known to form such kind of holoenzymes, right? So none of the other phosphatases are known to form such kind of higher order structures, right, higher order complexes. So this is where we went back and looked at the interactome again and then tried to see if there are higher order complexes that are formed by other phosphatases now, leaving alone the PPPs. So that's where we found these are known ones and these are the unknowns now. So you can see there are a lot of new communities that are being formed by these regulators with new phosphatases. They are not in the PPP family, but they are in other families now. So this is one example where we came up with, it is actually a functionally relevant complex that they are forming. So one example is that it's a phosphatase called ppm1G. So it's a nuclear phosphatase to start with. So what is known about this is it is known to regulate nuclear substrates and whatever functions that it does is through dephosphorylating nuclear substrates. In this work, what we have shown is that by binding to a different complex, so it's forming binding to a protein called B56 delta, which is a regulator now. So this is being shuttled into the cytoplasm, where this phosphatase is exposed to new set of substrates altogether. So there are nuclear substrates here now. With this complex, there are new set of substrates. So that the question that is answered is that their diversity is increased by complexing with a different protein. So the substrate that we identified is alpha-catenin, which is important for adherence junctions. right? So this is just one example. There are multiple of these new complexes that are formed, not in the PPP family, but outside the PPP family now. So we are currently expanding all those new complexes. So what I'll spend in the next 5 minutes is the last point where we wanted to address whether these physical interactions really translate into new functions, right? So for that, we ended up having a lot of new functions for this. I'm not going to touch on all of them, like vesicular trafficking, mTOR signaling, transcription, every possible new pathway. So the one which I'm going to talk about is the one where we entered into a new area, regulation of the histones, the histones and their modifications through phosphorylase. So I think many of you know that histones are subjected to various set of post-translational modifications, and phosphorylation is one such mechanism. But what is interesting is that the enzymes that are known to remove this phosphorylations on histones are very limited. I would say none. So the enzymatic machineries are not there. So given that we have the interactome available, so it's very easy for us to go back and see if there are any enzymes that we can assign to this modifications, right? So that's where we found these are different sets of phosphatases that are interacting with different chromatin-associated proteins now. Not only histones, but all the different chromatin-associated proteins. The one I'm going to talk now is a protein called SHP1, which is a tyrosine phosphatase. So it's a very interesting phosphatase. It's a phosphatase which is located in the cytoplasm, but it is interacting with the complex in the nucleus, right? So it's a complex called PAF complex, which is a subunit. There are 4 subunits of them: CD73, CTR9, LEO, PAF, and RTF1, along with the histones, right? So what does this PAF complex do is, it's an essential complex for transcription elongation. So what PAF complex is known to do is it binds to a polymerase, RNA polymerase, during transcription initiation at the first step of initiation. It recruits the E3 ligase called BRE1 RAT6 complex, the E2 and E3 enzymes, which ubiquitinates the H2B at a particular site at K120. And this ubiquitination is important for recruitment of a complex called FACT, which opens the histone octamer, right? So this ubiquitination is absolutely critical for the opening of histone, the chromatin, and the polymerase movement during transcription elongation, right? So it's critical that the PAF complex is very critical for the elongation step of the transcription. Since now we found that this complex and histones are bound, we wanted to see how this SHP1 is talking to this complex and the transcription machinery. So we asked what is the role of this SHP1 association with these chromatin proteins. First things first, we had to confirm that these are interacting specifically, and they all interact in the cell, the PAF components as well as the histone H2B. And this interaction is biochemically active. So when I say that, it is important for regulating the histone ubiquitination, which is regulated by PAF complex. So when you make a CRISPR knockout of this SHP1 in cells, you see that there is a drastic reduction in H2B ubiquitination. There is no H2B ubiquitination in these cells. And that is because due to the dephosphorylation of this H2B by SHP1, right? So what we went ahead and then tried to show is that what is the site of this dephosphorylation on histones, right? There are multiple tyrosine residues. Very interestingly, we found that there is a tyrosine residue at Y121. So please remember that the lysine is the one which gets ubiquitinated. So the 120 ubiquitination is happening. This is the residue where it gets ubiquitinated. So the tyrosine is sitting right next to the lysine, right? So what we showed is that when you mutate this tyrosine to a phosphomimetic mutant, so you can see that there is no ubiquitination. So what it suggests is that when there is a phosphorylation on this residue, ubiquitin is not able to attach to the lysine, so there is no ubiquitination. So that's exactly what is happening. So when you make this phosphopeptide, there is no ubiquitination, right? Mechanistically, how is it doing? So what we showed is that it is, we know that it is important for recruiting this complex. So we showed that it is important for binding of this E2 complex onto the histones, right? When you make this mutant which is hyperphosphorylated, you will not see the binding of this E2 to the protein, to the histones. So it's all good. SHP1 is doing dephosphorylation, it is regulating histone ubiquitination, but is it really going to the transcription sites, right? So it's a cytoplasmic, as I said, it is a cytoplasmic phosphatase. So it is cytoplasmic, but under conditions where transcription is active, it goes in, localizes the transcription machinery. You can see with the co-localization with RNA Pol2, CDK9, and the active RNA Pol2 as well. So if it is going to transcription, so what will happen if you deplete this or delete this in the cells? So it is going to transcription, and then it is enriched at the transcription initiation sites. And if you deplete this, when you make a knockout of this SHP1, so you see that there is a drastic reduction in the transcriptional initiation of this at the global level, right? So there is a reduction in the H2B-UB and as well as pausing of RNA Pol2 at the transcription machineries, right? So if you calculate the pausing index, in the cells where SHP1 is deleted, all the transcription machinery is paused at the initiation stage. So it's not able to move along the chromatin. So it's not very surprising that it reduces the elongating polymerase, accumulates the initiation stage only, right? So it's a global change, and you can see that there is a drastic reduction in the transcription, overall transcription in the cell. So in the knockout lines of SHP1, right? So I think I finished in time. So this is the model that we came up with for SHP1 role in transcription. So under normal conditions, what you have is a PAF complex bound to RNA polymerase. So it recruits this SHP1, which now acts as a switch for dephosphorylating the Y121 residue in the H2B tail. This dephosphorylation is absolutely necessary for the addition of ubiquitination onto the K120 site, which is now able to open up the chromatin, so which makes the RNA polymerase move along the transcription, right? So when you have the SHP1 deleted in the cells, there is no dephosphorylation, tyrosine remains phosphorylated always, no ubiquitination, RNA polymerase gets stuck at the initiation stage, there is no movement of transcription. There is no movement of RNA Pol2, there is no transcription. So this is the story that came up recently. So the take-home points from this work and the ones which I could not present also, so is that we believe that there are diverse unidentified cellular functions for phosphatases, which we continue to identify. And what is interesting is that these functions may not necessarily require its catalytic activity. There are a lot of functions by these phosphatases which do not require their catalytic, the enzymatic activity. There are a lot of enzyme-independent functions as well. And they assemble multimery complexes outside its PPP complexes to enhance the substrate and functional diversity, right? So that's where I land. I had to acknowledge the group. So this is the group that is currently in the lab. I had to acknowledge previous students as well. And thank you all. I'll take any questions.
Thanks a lot for sharing interesting piece of work and also, being accurate with time. So other questions? Manoj and Monica.
Just a quick query about the knockout cell lines you made. Any clue about the knockout animal, whether it has been made, and what do you think about it?
Yeah, so there is a SHP1 knockout animal. So they are thick, they are small. So they have inflammatory problems. So they have immunologically related problems. It's not embryonically lethal, but they survive for some time. They survive for some time and die at very early age. So they do survive for a couple of weeks. So that's it.
Yeah, Manoj.
Okay, thank you. Excellent talk. Congratulations for the same. I have a more general question, like the problem we have in targeting kinases for drug discovery development because of the ATP binding site and the common kinase domain. So what do you think, like with respect to so many phosphatases, and there are also non-specific and tissue-specific. So what is your take on like discovering and developing molecules which can target a specific phosphatase? What can be a strategy? Can you comment on that, please?
Yeah, sure. Yeah, this is a question that is frequently asked in the field or anybody who gives talk on phosphatase. So I do not agree that it is phosphatases are they're not promiscuous as everybody thinks, right? They are very specific, right? the approaches that people go after phosphatases, that they try to do the catalytic enzymatic inhibition, right? So they try to find inhibitors for enzymes. I feel that is not going to work, right? So because there are a lot of substrates, right? So the obvious approach that we think is going to probably take forward is the binding surfaces, specific binding surfaces for specific set of interactors. That's where we try to sell our story like that, right? So we believe that the interaction surfaces between different protein complexes are much better targeted, probably give more specificity, I feel. Yeah, so for example, if you look at the P10 WP2, right, I can cite that. So you know, we exactly know where they bind. So if you make we have a peptide which inhibits this binding. When you make this disruption, it's very active in tumor suppression, right? So we believe that it is important to have specific peptides or specific small molecules which can interrupt this interaction specifically. Yeah. Sorry? So, yeah, it's, I don't know. So it's a very difficult question to answer. So there are a lot of phosphatase there. I think there are very specific phosphatase for specific tumors. That is there. There probably you can try, but not every phosphatase in every tumor cell mostly. But for some hematopoietic tumors, they tried. This one is actually hematopoietic phosphatase, the SHP1. They try to do. That's the reason they come up with the the functions which are related to the immune related. Tumor specific, I doubt. So,
yeah, Deepak, last question.
Very motivational and inspirational too. Wonderful publications. Congratulations. Just I wonder, what is the clinical status of your SHP2 if you look back to the patient samples and everything?
In tumors, there are a lot of mutations in SHP1, but I don't think anybody explored in the tumor direction. They all are interested in the immunological related diseases, immunity related diseases, autoimmunity related.
Even in the TCGA database, did you?
There are a lot of mutations. We do not know what these mutations mean to the cell. So we are looking at the some of these mutations, which if they are able to control the transcriptional machinery or not. So if they are not able to translocate to the nucleus, whether they are defective in binding to the PAF complex or not. So that's going on. Yes.
Completely on.
Yeah.
Okay, so he's around for some time, I guess,
I'm
and
around.
so
Yeah.
people can contact him and ask questions. So we will have the final felicitation for the rest of the awardees. So I request you to stay here, and Ram and Alakesh can come upstairs, and I request Director and Professor Vijay Chandru to come on the stage. So Alakesh, Ram. Madhika. So thank you. Over to Smriti for closing of the session.
A very good afternoon to all of you. It's indeed a great privilege and delight to stand before this august gathering to propose this vote of thanks. First and foremost, on behalf of entire CDRI family, I express my deepest gratitude to our honorable chief guest, Professor Dr. Vijay Chandru, for accepting our invitation. Thanks a lot, sir, for traveling all the way to Lucknow and gracing this occasion. The talk that you delivered today was of immense significance to all of us, and I'm sure that our faculty and students will be greatly benefited from this interaction. I take this opportunity to also extend my deepest heartfelt thanks to all the awardees for their wonderful orations. Thanks a lot for sharing your findings and your opinions with us today. I thank our Honorable Director, Dr. Radha Rangarajan, for her constant support and complete involvement in each and every aspect of the event. Thank you, ma'am, for your valued inputs. On behalf of Organizing Committee, I expressed thanks to all the staff and personnel who helped us either directly or indirectly for making this event happen. And last but not the least, I want to thank all the audience who have gathered here to make this meeting successful. Thank you all. May I now request all of you to rise for national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे